Okay, awesome. Okay, everyone. Hi. Sorry, the technical difficulties here, but so tonight we're talking about autism, and I'm going to take this from a biochemical perspective because most people are taught what autism looks like, but not what's actually driving it. And that one sentence is the reason why I wanted to host this webinar. Because when we only focus on behaviors or labels or checklist, we miss the important question of all, which is what is happening inside of the body and specifically inside of the brain that leads to these patterns happening. So tonight it's not about changing someone, or we're not blaming or pointing fingers or coming up with different theories of what's going on. It's about understanding biology. So that what we see on the outside finally starts to make sense about what's going on in the inside. So for those of you that don't know me, I'm Dr. Catie Harris, and I work in functional and integrative medicine. And much of my work focuses on chronic conditions where conventional answers basically stop short, right? So conditions involving the brain, the immune system, and energy production, which we don't have a great methodology for in conventional medicine. And autism is one of those conditions, right? So tonight we're going to look at autism through a different lens, not as a random neurological condition, not as a mystery with no explanation, but as a pattern of biological stress, particularly involving a couple systems. So energy production in the brain, inflammation and immune signaling, and the integrity of myelin, which we'll talk about. And that's the insulation that allows brain cells to communicate clearly and easily. And when these systems are under strain, the brain doesn't just fall apart, it adapts. And many of the behaviors that we label as symptoms are actually adaptive responses to that stress. And so what if the brain is responding exactly as it should, as it's supposed to, given the environment that it's actually in? So that's what we want to look at today. So I will start this with a genetic question. So genetics, human genes have to evolve over thousands of years, not a couple of decades. And a shift from rare to common within 50 years cannot be explained by inherited genetics alone. So when we see autism rates rise as dramatically as they have risen in a single generation, it tells us something important, right? So genetics don't change populations at scale in 30, 40, or even 50 years. I mean, you're looking at, it would have to be thousands of years. So that rapid shift, something that happens over 50 years, requires an environmental and biological pressures acting on existing genes. So now you're talking about genetics versus gene expression, and they're two completely different things. So genes are the blueprints that you are born with. Gene expression is how the blueprint is turned on or off, for that matter. And what can change is that gene expression. That absolutely can be changed, but what is it influenced by? What changes gene expression? And that's stuff like inflammation, metabolic stress, toxins, right? Nutrient deficiencies, immune activation, mitochondrial dysfunction. And it's not just autism. There's lots of conditions that are affected by all of those things. Our genes haven't changed. How the gene expression is what's being changed, and that's environmental. So genes will load the gun, but the environment is pulling the trigger, if you want an analogy. So the genes may create that vulnerability, but biology and environment determines whether the vulnerability is expressed. So autism appears to involve a genetic susceptibility, but not a genetic destiny, right? Just because you have the gene doesn't mean it will be expressed. What's changing rapidly isn't our DNA, because that doesn't really change. It's the biological environment in which the DNA is operating. And that environment includes energy availability, inflammation levels, immune signaling, and the integrity of myelin and cell membranes. And one of the most common explanations we hear is for rising autism rates is increased awareness on broader diagnostic criteria. And absolutely, awareness has improved. There's no question about that. But awareness alone can't explain the magnitude of the pattern of the increases that we're seeing. So if this were primarily an awareness issue, we would expect to see a shift in who gets diagnosed or earlier recognition of the same underlying population or stable rates of severe and functionally impairing autism. But instead, we're seeing something very different. So we are seeing increases across all severity levels, including children with profound support needs, rising rates in very young children where overdiagnosis would be unlikely, and parallel increases in related biological conditions, including immune dysregulation, allergies, autoimmune diseases, metabolic disorders, and neuroinflammation. So awareness can change labels, right? But it doesn't create a new biological burden at scale. And awareness doesn't explain why autism is rising alongside other conditions that share the same underlying drivers, like mitochondrial dysfunction, chronic inflammation, and disrupted gut-brain signaling. These are problems across the board. They don't cause autism per se, but they are linked to a lot of different chronic conditions. So that tells us that we're not just recognizing more autism, we're creating an environment that places increasing biological stress on developing nervous systems. And that's a lens that we're going to use for tonight. So awareness may explain some earlier under-recognition, but it can't explain a 50-year shift from rare to common. In the 70s, an autistic child would be in the hospital, and everybody in the hospital, all the residents would come and they would do a special case study on that child because there was one physician, I can't remember his name off the top of my head, but he recalls having one of those conferences where the patient was autistic, and he brought in all his residents and said, "You may never see another case like this in your career, in your lifetime." That's 1970. That wasn't that long ago. But anyway, it's what we're seeing is not just better labeling; it's increasing biological stress on the developing brain. So, this conversation is for you if you've been told, this is just how autism is, but feel like there must be more beneath the surface. You or your child are struggling with regulation, sensory overload, fatigue, sleep, or focus, even with therapies in place. You've tried behavioral education or supportive interventions, but results feel limited or inconsistent. You notice gut issues, immune challenges, inflammation, frequent illness alongside neurological symptoms. Right? You just want to understand autism at a biological level and not just manage outward behaviors. And you're seeking clarity, not blame, and insight, not false promises. So if any of this feels familiar, you are in the right place. This webinar isn't about changing who someone is. It's about understanding why the nervous system may be under stress and how biology shapes what we see on the outside. And most importantly, what can we do about it? So I am going to share a video with you, and I have permission to share the videos and case studies. This is not my patient, just to be clear, and I had no involvement in this case. But what I want to highlight is the behavior and the change in behavior, and we'll talk a little bit about why the behavior changed.
This is a before and after for one of Dr. Radin's patients.
Nice job, bud. Are you beating Dad? Oh, look up, bud. Yes. Way to go, Shawno. Amazing. Fantastic. Was that fun, bud?
So a much different to go from head hitting and agitation to a greater calm is, I mean, that's pretty impressive, right? And, she mentioned prodrome glia, which I'll talk about in a little bit, but what prodrome glia is made up is our plasmalogens, which are the molecules that create myelin, and that's what we're going to talk about tonight. But when you see a shift like Sean's from head hitting to greater calm, the most important thing to understand is this. Behavior changes when the nervous system is no longer under constant biological threat. So in autism, when you have self-injurious or highly dysregulated behaviors, they're often protective responses, not intentional actions. So they commonly reflect overload, sensory, inflammatory, or energetic, okay? And we use plasmalogens, which are molecules that regulate brain stability. So let's break that down for a little bit. And we're going to talk about this in a couple different ways. So you don't have to remember everything like right this second, right? So just listen, and you can always go back over this. Glia cells are the brain's regulators. They're not just support cells. They regulate neuroinflammation. They control synaptic signaling, which I'll show you pictures of. They support myelin maintenance, manage the brain's immune system response, and they buffer excess neurotransmitter activity. And when those glia cells are stressed or inflamed, the brain becomes noisy, overactive, and poorly regulated. So they can show up as agitation, sensory overwhelm, or self-soothing behaviors like head hitting, right? So membrane and myelin stress is going to equal signal distortion. And glia cells are membrane-dense cells. So when membranes lack proper phospholipid structure, those signals will fire inconsistently. Sensory input feels chaotic. The brain is struggling to filter information. And when signaling becomes cleaner and more predictable, then the brain no longer has to compensate with extreme behaviors. So reduced neuroinflammation is going to lower that threat level of the brain as well. So chronic microglial activation, I have a picture of this, so keeps the brain in a defensive state. And that state looks like hypervigilance, right? Reactivity and poor emotional regulation. So supporting glia health helps shift the nervous system from I'm under attack to I'm safe enough to regulate. Okay, the body has to feel safe. Calm doesn't come from sedation. It comes from reduced inflammatory signaling. And energy efficiency improves regulation capacity. Okay, the brain is a very energy-intense organ. And when your membranes and mitochondria are inefficient, the brain's going to run hot, but underpowered. Its regulation fails under stress, and small inputs can trigger large reactions. And this is what you see in kids with autism, maybe your own kids, maybe your own, and this isn't just for kids, this is for adults too, that have grown out of childhood and they're adults with autism now. So the focus is the same, whether you're 2 years old or 22 years old or however old you are. By improving membrane function and glial efficiency, energy demands will start to decrease. The brain can stay regulated for longer periods of time. And that's when, your family members or caregivers will notice fewer meltdowns or less self-injury, faster recovery after stress. So in this video, when you saw Sean become more calm, it wasn't because the behavior was suppressed or he did behavioral therapy. It's because the brain no longer needed to defend itself in the same way. And that happens by supporting glia cells. So because the glia cells are the regulators of inflammation, myelin, and signaling, okay? We changed the environment that the brain was operating in. You got the glia, okay? The glia is a plasmalogen replacement therapy we'll talk about. And when the environment changes, behavior will often follow as well. So our goal here is not to, like I said, we're not here to change who someone is or assign blame, or we're not offering quick fixes. There is no quick fix for this. This is, a very serious condition. Our goal is to help you understand autism through a biological lens. So I'm going to use words that you might not be familiar with. Maybe you are familiar with them. But if this is the first time you're hearing them, it's okay if you're not understanding everything that we're talking about. But that you'll see behaviors will begin to make sense once you understand the biology behind it. So this can help you to go down another pathway when you're looking for answers. So we're going to look at how energy production, inflammation, myelin, and the gut-brain signaling shape the nervous system function, and why stress in these areas can show up as dysregulation on the outside. But most importantly, the goal here is to replace confusion with clarity. So support becomes thoughtful, right? Grounded and rooted in understanding rather than guesswork. So there's often a gap between receiving an autism diagnosis and understanding what's happening biologically. So families are given labels and strategies, but not always a clear explanation about why the nervous system is under stress in the first place. So for those of you that don't know me, I'm Dr. Catie Harris. I'm a nurse practitioner, integrative medicine clinician, and my work focuses on complex chronic conditions where conventional answers often fall short, especially conditions involving the brain, the immune system, and cellular energy. So I work with a small group of elite practitioners with Dr. Goodnow, who is a world-renowned biochemistry expert, amazing man. All of this information is coming from my studies with him and the practitioners that work with him as well. So he's teaching us how to bridge biochemistry into clinical medicine. Very, very powerful because it's sorely lacking, right? We do a lot of clinical medicine that's based on population health, but not cellular health, right? And the biochemistry research is extremely rich. The videos and stories that I'm going to share tonight about autism come from the dedicated work of my peers, and I'm very grateful to share them with you to help you understand what is happening. So over the years, I've worked with families and patients navigating autism, neuroinflammation, and metabolic stress, and I've learned that behavior makes a lot more sense when you understand the biology beneath it. So, and again, I want to be clear about how I'm approaching tonight's conversation. I'm not an autism specialist in the traditional sense. I do not focus on diagnoses and labels. My specialty is cellular health and membrane health, specifically membrane integrity and mitochondrial function, hence, my YouTube channel. And how these disruptions in the systems affect the brain, the immune system, and the overall regulation. So autism is one of those conditions where we see biological patterns show up very, very clearly. So tonight, I'm not here to speak about autism as a diagnosis. I'm here to share a framework for understanding what's happening at that cellular and metabolic level, and why supporting membranes and mitochondria can change how the nervous system functions. So with that lens in mind, membranes, mitochondria, cellular resilience, let's step back for a moment and look at the bigger picture, because before we talk about strategies or support, we have to understand the pattern. Now, autism isn't something that appears out of nowhere, okay? It reflects a nervous system that has been operating under increasing biological stress. To see that clearly, we need to start with what's changed. And one of the most striking changes is how common autism has become. So I want to share a brief case that illustrates not what I mean when I say that behavior often reflects biological stress and not intent. So this is a 5-year-old boy with autism whose parents described significant dysregulation. Before making any changes, he struggled to sit calmly in public, was mostly nonverbal, and had difficulty with focus, independence, and just sensory processing in general. So everyday environments were just completely overwhelming for him. Then he started plasmalogens, which we are going to get to, but his parents noticed something very important. This is like Sean, the video that we just saw. They didn't see a dramatic personality change, but they saw a shift in regulation. So he became noticeably calmer. And for the first time, he was able to sit quietly in a restaurant. I know families that haven't been able to go to a restaurant for years. So this is not a small thing. So he began noticing things in his environment, like stopping to smell flowers, which is a sign that sensory input was no longer overwhelming his nervous system. What was especially interesting is that the people around him noticed these changes without even being told that anything had changed. So his aide commented on how much more engaged he seemed. And over time, he went from being mostly nonverbal to speaking small, short sentences daily. He began eating more, listening better, and showing new independence, putting on his own shorts, riding his scooter, and engaging socially in ways that he hadn't before. So from a biological perspective, this actually makes sense because when the brain is overwhelmed by inflammation, poor energy efficiency, and impaired signal insulation, it struggles to filter information. So everything feels loud, everything feels urgent, and calm becomes virtually impossible. So by supporting those glia cells, the membrane integrity and myelin, the system that helps regulate the brain signaling, the nervous system becomes more quiet and more efficient. And when that happens, regulation improves first and behavior will follow second. So this wasn't about suppressing behavior. It's about reducing the biological load on the brain so that it can finally do what it's designed to do. So I do want to show you the biochemistry of autism, and we're not going to shy away from the biochemistry of it since it's a biochemistry lecture, but you don't have to repeat or understand everything in order to get something out of this. This is for educational purposes. We're not intending to diagnose or treat any disease tonight, but at the same time, I want to share with you that what we know in autism is a lot. Biochemistry knows a lot about this condition, and that's what I want to share with you tonight. So what we'll talk about is autism reflects biological stress, not broken brains. Energy, inflammation, and myelin determine how the brain processes the world, and supporting membranes and glia cells changes the brain's environment, not the person, and that's what we're looking to do. So to understand autism, we have to start by letting go of one common assumption that the brain is malfunctioning on its own. The brain doesn't operate in isolation. It is the most energy-dependent organ in the body. It is exquisitely sensitive to what's happening biochemically everywhere else. Every thought, every movement, every moment of regulation requires energy. Just sitting, listening to me is requiring energy, a lot of energy. Intact cell membranes and tightly controlled inflammatory signaling. So when those systems are under stress, the brain doesn't fail, it adapts. And from a biochemical standpoint, what we often see in autism is a state of chronic metabolic load. And that load can come from a lot of different places. It can come from mitochondrial inefficiency, immune activation, oxidative stress, gut-derived inflammation, right? But the result is the same. So the brain is being asked to do too much with too little. So let's talk about energy for a moment. Neurons rely almost entirely on mitochondria to generate ATP. What's ATP? That is the body's energy currency. That's how you run, right? Like gas in a car. Your gas is ATP. So when mitochondria output is inefficient, the brain becomes energy constrained. Okay, that an energy constrained brain cannot regulate smoothly. It prioritizes survival over refinement. And that can look like reactivity, rigidity, sensory overload, shutdown. Think about it. If, when you're under a lot of stress and you have people coming at you all different ways, you're not in your most refined moment. You might snap at people, like you might become dysregulated yourself. So at that same time, inflammation changes how signals are transmitted. Inflammatory molecules alter neurotransmitter balance. They'll increase background noise and activate glial cells into a defensive state. So when glia cells are chronically activated, these are the cells in the brain remains on high alert, even in safe environments. It can't calm down. And then there's, of course, the membrane piece. So every cell membrane, every brain cell is wrapped in lipid-rich membranes, okay? And these membranes aren't just structural. They determine how signals move, how receptors respond, how efficiently the cell uses energy. When membrane composition is compromised, then the signaling becomes distorted and metabolically expensive. And this is how your stem cells and your peptides would work. They are signaling. They're signaling the molecules. But if the membranes aren't intact or that composition is compromised, they're not going to work, or they're not going to work as good as they could. So when we say autism reflects biological stress, we're not talking about a single pathway or a single cause. We're talking about the biochemical environment where energy demand is high, inflammation is persistent, and signal transmission is inefficient. In that environment, the brain does exactly what it's designed to do. It adapts. Behavior in this context is not the problem. It is the output of a nervous system working under load. So I want to take a few minutes to explain something that fundamentally changes how we interpret behavior, especially in autism, because behavior is it's not a personality flaw. It's not a choice. It's a reflection of biology in real time. So let's start with energy. The brain consumes about 20 to 25% of the body's total energy. Okay. You need to feed the body the energy that it needs in order for it to get that energy. The brain itself only weighs 3 pounds, and it's only 2% of the body weight, but it takes 25% of all of your energy, right? It's like the mafia coming in and taking all your money. But every function, attention, emotional regulation, language, sensory filtering, it all depends on adequate ATP production inside the neurons. So when mitochondrial output, the mitochondria, remember, is making the ATP, when it's inefficient, then the brain's got to conserve energy, right? If you are running low on food and there's a famine, you're not going to eat everything today and have nothing tomorrow. You're going to conserve that energy. And so the brain's the same way. An energy-constrained brain cannot multitask, right? It cannot filter noise efficiently. It cannot regulate smoothly. So it simplifies. And that simplification can look like repetitive behaviors, rigid routines, reduced verbal output, right? You don't have enough energy to speak. Or it could look like withdrawal. So not because the child can't, but because the brain is prioritizing survival over complexity. Now layer inflammation on top of that. Inflammation is not just swellness or redness like we think of, you sprain your ankle, it gets red, that's inflammation, but it's also a signaling state. Inflammatory cytokines actively change how neurons fire, how neurotransmitters are released, and how much background noise exists in the nervous system. So inflammation raises the baseline signal in the brain. So imagine trying to hear a whisper in a room where there's 10 radios that are blaring, right? And they're all playing at once, and you're trying to hear one voice. That's what inflammation does to neural processing, okay? It amplifies noise and it reduces clarity. So this is where immune signal becomes critical. So the immune system and the brain are in constant communication. They're always talking to each other. When the immune system is activated, whether it's an infection, it could be gut inflammation, it could be environmental triggers, it could be chronic stress. It's going to send, the immune system's going to send chemical messages to the brain, and the brain has to be able to hear them. Those messages are going to tell the brain to shift into protection mode. In protection mode, the brain becomes more reactive, less exploratory, and more sensitive to sensory input. It's not a dysfunction. It's an adaptive survival response. It allows that person to continue living, right? Because that is the brain's priority, is to continue to stay alive. And it's under such pressure that these are the choices that it has to make. So you might not like the choices the body makes for you, but it's making that choice because it wants to survive. So here's the key point. When you combine low energy availability, ongoing inflammation, and persistent immune signaling, the brain exists in a state of a chronic load. And in that state, behaviors are going to emerge that help the nervous system to cope. So stimming to regulate input, avoidance to reduce demand, aggression or shutdown when capacity is exceeded. So behavior is not the starting point. Behavior is the nervous system saying, this is too much. And once you understand that, the goal shifts and we stop saying, well, how do we stop this behavior? And we need to start asking, what is the brain responding to and how can we help reduce the load? All right, so this, again, it's not my patient, but I did want to read a little bit about this to you. So this is a patient, she was normal birth, had all her immunizations, but started with developmental delays around 9 to 12 months. such as not walking, not responding consistently, poor eye contact, not saying any, mama or papa. She wasn't communicating. When prompted, she would clap her hands and sit down, but they were the only commands that she seemed to understand. She did begin walking at the age of 18 months, but her development had already slowed dramatically in all other areas. She became socially indifferent, made no eye contact, didn't respond to her name, didn't understand routine instructions. So maybe some of you can relate to this. So, and they saw one of the providers, one of the elite practitioners for the first time, and they started glia, which is the prodrome glia, which is for the glia cells, right? That's the myelin. So basically it's providing plasmalogens and building up the myelin, which we'll talk about. We'll get to that. So she started on a fairly low dose. And again, prior to initiation of those plasmalogens, she really wasn't able to do much, had 4 to 6 words, maybe 10 to 15 receptive labels. But afterwards, there was no response to her name or eye contact. So she just was unable to follow routine commands. So after 2 months on the glia, so Sean in the previous video was 1 week. This is 2 months after glia. And she started to retain information, had expressive language. She could name the colors and shapes and fruits and could recognize numbers up to 25. She could follow one-step commands. She could understand basic spoken language, able to point to things that she wanted. Her eye contact improved. She could jump, dance, hug, wave. I mean, these are things that are so amazing. And they might seem little to somebody that isn't as familiar with autism, but they're absolutely, absolutely huge, and for the families as well. So what we call a behavior is simply the brain adapting to its internal environment. Energy deficits limit capacity, and inflammation increases noise. So before a brain can regulate behavior, communicate, or learn, it has to have enough energy to do those things. So the brain is the most, again, energy demanding organ. And even at rest, it's using 1/4 of our total energy. So when it's really working, it's using even more. So every skill that we care about, our attention, language, emotional regulation, sensory processing, they all require energy at the cellular level. And when energy production is efficient, then the brain has flexibility and it can adapt, it can shift attention, it can tolerate stress, it can recover. But when energy production is impaired because your mitochondria, which are inside the cell, if they're not working efficiently, the brain has to conserve. And when the brain conserves energy, it simplifies. And again, the simplification is what we call behavior. The reduced speech, the repetitive actions, the rigid routines, withdrawal or explosive responses. These aren't signs of the brain failing. They're signs of the brain operating at the edge of its energy budget. So at a biochemical level, this comes down to mitochondrial output, right? So if the mitochondria can't generate ATP efficiently, then the neurons can't maintain firing patterns. So first and foremost, autism is a mitochondrial dysfunction. It's a disease of mitochondrial dysfunction. Without enough ATP, the brain loses its ability to multitask, filter sensory info, and regulate emotions simultaneously. So regulation requires extra energy. So the first thing to go when energy is limited is regulation. So that's why we can see that a child can perform well in one area, but struggle in another. It's not inconsistency, it's a capacity issue. So energy doesn't determine your intelligence. It just it determines your capacity. And when energy is limited, the brain becomes more vulnerable to its environment, which brings us to inflammation and signal noise. So just looking at this slide, for many of you might say everything, right? But once energy becomes limited, the next factor that shapes behavior is going to be inflammation. So inflammation in the brain doesn't look like a fever or swelling, like I said, like a sprained ankle. It shows up as noise. Inflammatory signals, cytokines, oxidative stress, immune mediators, these change how neurons fire and how messages are transmitted. So they raise the baseline activity in the nervous system. When baseline activity is high, the brain loses contrast, right? Everything feels loud, intense, and urgent. Again, it's like trying to listen to that single voice in a room where there's 10 radios playing all at once. The voice is not gone. It's still there. It's just that the noise is too high and you can't focus on it. So in the brain dealing with inflammation, sensory input becomes overwhelming. Lights feel brighter, sounds feel sharper, touch is going to feel threatening. The nervous system shifts from thoughtful processing to rapid reaction. And this is where your immune system matters. The immune system communicates directly with the brain. And when it's activated by infection or gut inflammation or environmental stressors, oxidative stress, it's sending messages that tell the brain to move into protection mode. Protection mode is not dysfunction. It is survival biology. Again, you might not like the way the body reacts, but in order to have energy, you need to consume energy and good energy, right? And then you have to be able to process that energy, and that's where your mitochondria come in. Inflammation doesn't change who a child is. It just changes how much noise their brain has to process. So now if you have a brain with limited energy and increased noise, you're gonna have behaviors that reflect that. So the final question becomes, well, what determines whether that brain can filter noise and make sense of the world? And the answer to that is myelin. Okay, that's what we were talking about before. If you didn't know what myelin was or is, that's what we're going to talk about. So now that we've talked about energy and inflammation, which is capacity and noise, we can come to the structure that determines whether the brain can make sense of it all, and that's myelin. Myelin is that insulating layer around the nerve fibers. It doesn't create signals. It makes signals precise. So think of the brain as an electrical system, right? Without insulation, signals are going to leak or overlap or they interfere with each other. Myelin is that insulation, and it determines how quickly signals travel, how accurately they arrive, and whether the brain can separate what matters from what doesn't. A brain with healthy myelin can filter background noise, can focus attention, can coordinate speech and movement, can regulate emotion, and learn from feedback. But a brain without sufficient myelin cannot do those things reliably, no matter how hard it tries. Because learning is not just neurons firing. Learning is about signals being transmitted consistently, enough to be reinforced. So in autism, you have a myelination impairment, right? We call it demyelination. So it's either impaired or delayed, and the brain is struggling to stabilize the signals. That instability comes up as difficulty filtering sensory input, challenges with attention and impulse control, disrupted communication, and troubling integrating social cues, not because the brain is incapable, but because the signals aren't clear. And a brain that can't filter noise can't regulate behavior. And a brain that can't regulate can't learn. So the good news is that myelination is not a fixed process. It's an ongoing, energy-dependent, and biologically supported process, meaning, when we reduce inflammation and support membranes, signal precision can improve. And that's why membrane supports matters, because when myelin improves, the brain doesn't have to fight itself just to function. So even with enough energy and lower inflammation, the brain still needs a way to process information accurately, and that's myelin. So why does this matter for regulation? Well, regulation requires timing and precision. So emotional regulation, attention, speech, motor control, these all depend on signals arriving in the right order, at the right strength, at the right time. If the signals arrive late, scrambled, or inconsistently, the brain can't coordinate the responses. So when you have myelination that's impaired or delayed, the brain is going to struggle to filter noise. So everything feels equally important, right? You can't give weight. Everything feels intense and nothing is fading into the background. So again, that's where you see behaviors like sensory overwhelm or difficulty focusing, the impulsivity, not because the child's not trying, it's because the brain can't separate signal from noise. without signal precision, the brain literally just can't filter the noise. And without filtering it, it can't regulate. And without regulation, learning is going to stall. So now babies are born with a deficiency in myelin. Okay, we know this. The myelin starts to come and myelin occurs. Your brain is myelinating up into your 50s and 60s. Like it myelinates for a long time. But because they're not born with a whole lot of myelin, it's a very, very small amount of myelin, they have to create myelin and they start. They get some myelin from the mother in the form of plasmalogens. So plasmalogens are molecules that make up a large portion of the myelin. So the mother gives the baby all of their plasmalogens to myelinate a little bit, right, up until about 30 weeks gestation. And around 30 weeks, the baby starts to make its own plasmalogen so it can myelinate its own brain. So that at birth, the baby is very deficient. That's what you see in these MRI pictures. So, but when at birth, the baby is still deficient, but will continue to breastfeed and make its own. That's how it's getting its plasmalogens. That's how it's getting. That's how it's myelinating. So, as the baby learns new things, new myelin is created, and that corresponds with white matter imaging. So, 3 months they're turning their head, 6 months they're responding to other people's emotions, and the myelin is creating that connection between neurons and providing that signal precision so that the baby can do these things. And you can see the red, right? As the red is the myelination. So from at 19 weeks to 34 weeks. The myelination is becoming increasingly more and more dense. 34 weeks, more dense. They're learning to do more and more things. They're making these connections. And myelin will increase into adulthood, and it'll peak around the age of 50 and then potentially decline, which is another lecture on the opposite end of the spectrum. But that myelin is really, really important in order for all of these milestones to be met. So when your child goes for a milestone check, what they're really doing is assessing myelin production, even if your provider doesn't realize that's what they're doing. So white matter maturation profiles predict cognitive abilities. And we've known this for a very long time. This is something that we've seen. But why? So myelin is made in the brain by cells that are called oligodendrocytes. Okay, remember, you don't have to remember all of this, but their purpose is to spin myelin. And they spin it around the nerve as the person learns new things. So you learn to play tennis, right? You start myelinating those axons so that you can play better and better. And someday you become Roger Federer, right? But so he's probably got really strong myelin, axons for his tennis playing. But the myelin sheath is what we call the white matter of the brain. And the white matter connects the gray matter. So the gray matter are your neurons. And the white matter, as you see here, is going to be all that myelin, right? So this is a bundle of myelinated axons. And on an MRI, that's what we see as white matter. So if you're looking at an MRI and you see some of it's gray and some of it's white, that white matter is all your myelinated axons. So a connection needs to be made between neurons in order for them to communicate. So this is what happens when you, again, learn something new. So when a toddler tries to walk, he doesn't have that neuronal connection yet, right? He's uncoordinated. He can't get it quite right, but he keeps trying. And so an axon connects those neurons that are required for him to learn how to walk. And then myelin starts to wrap around that axon to reinforce the skill, that learned skill. So this is true for learning anything, languages, how to cook, how to play tennis. The more you learn, the more axonal growth you have, the more myelin you need. So what is this myelin stuff anyway? What is this myelin sheath made out of? So myelin, like all cells, you have 40 trillion cells, but they're all made up of phospholipids. And the composition of the phospholipids changes somewhat from cell to cell, but the basic structure remains the same. So you get this weird looking phospholipid bilayer, excuse me. The bilayer is what makes up that sheath. So there's these molecules called plasmalogens, and plasmalogens account for over 50%, which is the major portion of phospholipids in the human brain, and especially in myelin. But newborn babies only have about 7% of plasmalogens. So they need to catch up because it's the plasmalogens that are making the myelin. Okay, the plasmalogens are the rate limiting nutrient for myelin development. Brain plasmalogens double during the last 8 weeks of pregnancy, and then they double again in the first 6 months, which is why you see such explosive growth and changes, from newborn to what a 6-month-old can do. And what is the source of plasmalogens? Well, breast milk is one, right? That's a big one. So as well as babies are able to make their own. Now, a caveat here, cow's milk, so any infant formula, there's no plasmalogens in milk. So the baby is completely reliant on themselves to get all the plasmalogens that they need. Babies that are breastfed for the first 6 months are at a distinct advantage for developing myelin. Now, having said that, humans are very diverse. Okay, some children will have naturally good plasmalogens, and it won't matter if they breastfeed or they don't breastfeed, right? So they can make their own, and it's not a problem. And there's certainly breastfed babies who don't get enough plasmalogens from the mother, because the mother can also have a plasmalogen deficiency. And indeed, older mothers do tend to have fewer plasmalogens. So if the baby does not get or make enough plasmalogens in those first 6 months, myelin development can be delayed or impaired. And this has been extensively studied. It's not controversial. It is very well established. So, does breastfeeding alter early brain development? And they've done large epidemiological studies that associate that breastfed children exhibited increased white matter development in later maturing frontal and associated brain regions. That's well known. Case control study, ASD was found to decrease in a dose-dependent fashion over increasing periods of exclusive breastfeeding. So the study indicates that ASD risk is generally associated with suboptimal breastfeeding practices. Another study in the first 6 months of life and autism spectrum showed that in robust multiple sensitivity analysis that toddlers without breastfeeding for the first 6 months had higher odds of autism. And it's because of the plasmalogens. And again, association of breastfeeding in the first 6 months of life, the prevalence is statistically higher. That doesn't mean for an individual in particular, whether you were breastfed or you weren't breastfed. Again, the human brain and the human body is very resilient. And if the child himself has the ability to make plasmalogens, then the breastfeeding won't be an issue. But statistically, the breastfeeding babies have lower incidence of autism. All right, so can we ask what causes autism? And the answer isn't to point fingers at anyone and blame anybody about anything, because the cause is impaired myelination that can result in autism, and it's inflammation, right? Neuroinflammation. So that destroys myelin faster than it can be replaced. And when you destroy myelin faster than it can be replaced, you're going to have delays, and you're going to have stalling in learning. You have inability to regulate behavior. So whatever the external trigger is, unresolved and chronic inflammation, neuroinflammation in a plasmalogen poor environment is how neurodevelopment is impaired. And that's what this study, again, there's been extensive research in biochemistry over the last 50, 60 years, even the last 100 years. Like plasmalogens have been known for about 100 years. They've known about them. They've been in thousands and thousands of research studies, but they're all in biochemical research studies, which is very, we just need to bridge it into clinical medicine because it's a really important concept. But the active inflammatory, neuroinflammatory process was seen in 100% of children with autism. So we know that microglial activation, neuroinflammation is a key aspect, regardless of what causes it. This is what we're dealing with, and this is what we need to do to make a difference. So let's go back just a little bit to the diagnosis of autism and pull this together. So, what is the diagnosis? The diagnosis of autism in the first year of life, there's usually no clear discriminating features. So you have to kind of go by the parental concerns. But around age 2 or 3, if they're having communication impairment, especially comprehension, unusual use of language, poor response to name, deficient nonverbal communication, and the absolute indicators for referral: no babble, no pointing, no gesturing by 12 months, no single words by 18 months, no 2-word spontaneous phrases by 24 months, and any loss of language or social skills at any age. Social impairments, lack of imitation, lack of showing with toys or objects, lack of interest in other children, odd approaches to other children, minimal recognition or responsiveness to other people's happiness or distress, or limited variety of imaginative play, especially social imagination, and impairment of interest, activities, and other behaviors. So oversensitivity to sound or touch, motor mannerisms, biting, hitting, aggression towards their peers, oppositional to adults, overliking for sameness, and inability to cope with change. Or repetitive play with toys, such as lining them up, turning light switches on and off, regardless of scolding. And so one notable interest is the high prevalence among boys, right? The ratio of autism from boys to girls is 4-to-1. And indeed, when girls acquire autism, it's usually much, much more severe than boys. And the controlling factor here is going to be estrogen, right? Estrogen provides protection for girls. And in fact, we see it later in life, like on the opposite end of the spectrum, when women lose estrogen during menopause, we see a progression of demyelinating conditions such as multiple sclerosis. It's the same concept, right? The myelin is impaired in multiple sclerosis. So, why don't adult women, acquire autism? So autism isn't something that gets turned on in adulthood because that developmental window, if you will, it's been closed. So in young children, their brains are much more susceptible because of the lack of myelin. Remember, the babies don't really form a lot of myelin up until like the first year of life. So they're working really hard towards it. But by the time you're 20, you have sufficient myelin. You can afford to lose some. You can, the myelination process has made connections, the proper connections that it needs. So in young children, again, their brains are much more susceptible because of the lack of myelin that already pre-exists. It's a pre-existing condition. So we can see autism as basically an opportunistic condition for unresolved neuroinflammation. So that unresolved neuroinflammation before myelin can be affected properly makes that a higher risk during the developmental years. But once the axons have made that connection and they've been properly myelinated, the myelin is continuously reinforced, and it's much harder to undo it because there's so many layers that are being made. Neuroinflammation later in life can cause injury, but it doesn't completely unmyelinate an axon or lose its connection, or at least to a scale that would cause an autism state. But young babies lack the connection completely. So that's a whole different story, right? It's one thing to repair a bridge, but if there's no bridge at all, that's a much different situation. So it has to be that connection has to be made first, and then neuroinflammation can prevent that connection from ever happening. So that is why autism is not a straightforward problem with a single solution. And it's when neurons can't communicate because of a lack of myelin, which makes up collectively your white matter again. These are the symptoms that will occur. So the neuroinflammation actively impairs myelin. And what causes neuroinflammation, right? Viruses, colds, head injuries, mold, stress, heavy metals, gut inflammation, mitochondrial dysfunction, pesticides, pollution, all of them, some of them, none of them. Like there's just so many things that can cause neuroinflammation. So any and all of these things can contribute. But the plasmalogens protect myelin, they create the myelin. So a rich supply of plasmalogens is required. So the question is, are plasmalogens enough to protect the brain? And to answer that, we move into our animal models. So animal models have been used for decades or forever, to simulate conditions. And it was found decades ago that the toxin, which is called cuperzone, predictably destroys white matter 100% of the time. So they perfected this model because, if you give too little cuperzone, there's not enough demyelination. And if you give too much cuperzone, then it kills the animal because you have a complete loss of plasmalogens in the blood. And ultimately, that's the cause of all death in humans is a lack of plasmalogens. Anyway, what they found was that if they gave a dose that was precise, they could induce demyelination 100% of the time in 100% of the animals. And this became the standard for autism studies, as well as multiple sclerosis and other demyelinating conditions. So what's of interest here is the question of the plasmalogen levels. So what happens to plasmalogen levels in demyelination? Well, they plummet predictably. So if you give cuperzone, right, which is a demyelinating toxin, then your plasmalogen levels will plummet. If you stop the cuperzone, then the levels will start to come back up, but they don't come back up fully to baseline, or not always. So Dr. Goodnow, who's the biochemist that I work with as an elite practitioner, he developed these plasmalogen precursors. So in 2006, it wasn't that long ago, okay? And the question he pursued once he made that was, if the animals were given the cuperzone followed by plasmalogens, would they recover? And the answer was a resounding yes, repeatable, just a completely repeatable study. And many of the animals not only returned to their original plasmalogen levels, but they exceeded that baseline. So the point is that, and also the other thing they did was they pumped up the animals with plasmalogens and then exposed them to cuperzone. And there was no effect whatsoever because the extra plasmalogens protected them from the cuperzone, which is amazing because we live in a toxic world. You cannot escape toxins. It's not possible. You just have to be able to manage the assault of daily toxins and to just be healthy, right? And to do that, you need a healthy supply of plasmalogens. So in autism, that assault of neuroinflammation is causing the demyelination. And there are many reasons the neuroinflammation started, but foundationally, the lack of plasmalogens is what allows it to continue and to remain unresolved. So if there's one message, from today, it's this: the brain doesn't change by forcing behavior; it changes by stabilizing the infrastructure, and that infrastructure is made up of membranes and glial cells. And every signal in the brain depends on membranes. Membranes control energy production, the immune signaling, the neurotransmission, and the inflammation. So if membranes are unstable, everything downstream becomes unstable as well. The energy, the signaling, the regulation. So glial cells are not passive support cells. They regulate inflammation and they manage energy distribution. They maintain myelin and they control how neurons communicate. So in children with autism, glial cells are going to be overactivated. They're responding to chronic stress, immune signals, or metabolic strain. And when glial cells are inflamed or dysregulated, the brain becomes noisy, reactive, and inefficient. And the opposite is true. When they're supported, the brain is calm, clear, and more adaptable. So, that's why traditional approaches focusing on correcting behavior aren't always, they don't restore biological capacity. So there's a piece that's missing there. You still need behavioral correction. And because autism is such a complex condition, there's not one thing that's going to change everything. But these things used in combination are going to be extremely helpful, but you're going to have a hard time correcting behavior if you don't correct with the biology of what's going on the inside. So support the brain, and the behavior will follow. Now, this is a little bit of a segue, but I think it's important, right before we get to the other stuff that I want to show you. But what a person eats is going to be very important too, because gut health is essential in autism, and it impacts neuroinflammation. So many kids with autism have gut issues such as chronic constipation, diarrhea, alternating constipation and diarrhea, excessive gas, bloating, burping, vomiting, all that kind of stuff, food refusal, very limited food choices. So behavioral symptoms can largely be driven by gut issues, which can cause increased irritability, aggression, anxiety, meltdown. There's that gut-brain axis. It goes, 90% of, what's going on in the gut is affecting the brain, and it's like 80% is efferent, which means that the gut is sending signals to the brain, and not on the reverse. It's only 20% of the time the brain sending it to the gut. So the gut's inflamed, you got issues. And myelin is not just a brain issue. It's a nutrition-dependent, gut-influenced structure. Myelin is built from dietary raw materials. So myelin is a lipid-rich, membrane-dense tissue. And if the raw materials are not available or they're not absorbed, then myelination is going to suffer. So myelin is mostly fat, but not just any fat. It's built from highly specific lipids that must be synthesized, transported, and assembled correctly. And they depend on essential fatty acids, the phospholipids, plasmalogen precursors, cholesterol, and adequate protein and micronutrients. So the diet lacks these components, or the gut can't absorb them, then the brain cannot build or maintain myelin efficiently. So the gut determines whether nutrients reach the brain, and it's not just what you eat, it's whether you can digest, absorb, and metabolize. So many children with autism don't have a supply problem, they have a delivery problem. So common gut issues that interfere with low stomach acid, poor fat digestion, dysbiosis or chronic inflammation, increased intestinal permeability, impaired bile flow, food restriction due to sensory overwhelm. So when the gut's inflamed, the brain is indirectly deprived, and gut inflammation drives brain inflammation. That's a critical bridge. So because the gut and brain are in constant communication, and an inflamed gut sends danger signals to the brain, and the glia responds by shifting into protection mode. So you can see how that's working. So myelin is especially vulnerable to inflammation because myelin is one of the most energy demanding structure that the brain builds. So when inflammation is high, again, the brain prioritizes survival, not maintenance. And sometimes restrictive diets can actually worsen the problem. So many families start removing foods to reduce symptoms, and sometimes that helps in the short run. But highly restrictive diets can unintentionally deprive the brain of myelin building materials. So you need animal-based phospholipids, cholesterol, fat-soluble vitamins, and adequate protein. The goal is not just to remove the triggers, it's to restore capacity. So, and when we support membranes systematically, we're not just helping the brain, we're helping the gut lining and the immune cells and the mitochondria, because they all require those things as well. So it's improvements will show up as better sleep and digestion. So you can't build clear brain signals on a compromised gut. So when we're talking about supporting membranes and glia cells, we're also talking about the entire gut immune brain axis. That's why it's not just one thing that solves everything, because there's a lot of things that need to be addressed simultaneously. Just a couple myths here. Autism is not caused by one bad food. It's a cumulative biological stress. Removing food is not going to automatically heal the brain, but the brain does need, requires those raw materials, especially lipids, cholesterol, protein, because they're needed for the myelin in the membranes. Plant-based or clean diets are not always better because plants don't have lipids, right? The brain is lipid dense. It needs lipids. So many critical myelin components are hard to synthesize if you don't have animal-derived phospholipids or equivalent precursors. If labs look normal, then nutrition isn't an issue. We all know that's a silly statement. And gut issues are separate from brain functions, which is not true. So the gut controls absorption, immune signaling, and inflammation, and a dysregulated gut is going to directly impair myelin. And just a word on omega-6, because it's everywhere. Omega-6 is a structural fat. It's not a bad fat, but you don't need nearly as much omega-6 as our diet allows. So any kind of seed oils, like your corn, soybean, canola, sunflower, safflower oil, any kind of reused cooking oils, ultra-processed foods, basically anything in a box or a bag or something sitting on your counter that doesn't rot is invariably going to have linoleic acid, omega-6, which can cause significant issue in the gut. So balancing omega-6 is going to be really important. So restoring membranes, myelin and brain signaling depend on healthy membranes. Membranes require dietary lipids, not just fat avoidance, and omega-6 is structural and necessary. Gut health determines nutrient delivery and immune signaling. Damaged cells drive glial activation and inflammation, and rebuilding membranes restores signal clarity, regulation, and learning. All right, so how can we what can we do with these issues, right? So I want to walk you through plasmalogen replacement therapy, and that's what we do with our patients. And not just autism, as I told you before, I'm not an autism specialist. I work with cell membranes, and autism has a root cause in membrane and mitochondrial dysfunction. So I would suggest you keep working with your current autism specialist or pediatrician, but do your research on what I'm going to show you now. And this is, again, not just for children, and it's not too late for anyone to start plasmalogen replacement therapy. So even adults with autism can benefit, and even people without autism can benefit, because there's a lot of chronic diseases out there, and plasmalogens are a huge part of health and wellness in general. So children are more resilient and see faster results, but everyone's brain has a certain level of resilience, right? And I'll share some videos of the work of my colleagues have done with autism, so you don't just have to believe me, you can just listen to the stories. So you can start your research at prodrome.com or on Dr. Goodnow's website. I'm an elite practitioner with Dr. Goodnow, and I believe in his products. I also take his plasmalogen therapy every day myself for better health and wellness. Now, remember, autism is first and foremost glial-driven neuroinflammation. So think of prodrome glia as a fire hose that is dousing the house with water. You aren't going to start rebuilding if the house is on fire. You need to calm the situation down first, and that's what prodrome glia does. Omega-9 is what builds myelin, okay? The plasmalogens and omega-9 are what builds the plasmalogens. So you can get prodrome from prodrome.com. If you use that coupon code, you get, I give all my patients a big discount with it. So now if you do some research, there's other products out there that claim to be plasmalogens, but Dr. Goodnow patented these in 2006. He's the one that's invested millions and millions of dollars into this technology. So, you know, the plasmalogens are exquisitely fragile and they're destroyed in the gut. So you can't just take plasmalogens. You have to take precursors, kind of like you can't just take dopamine. You have to take L-dopa because the molecule itself gets destroyed very easily. So don't waste your money on things you find on the internet. Just go to prodrome.com. Dr. Goodnow has authored most of the papers that are out there on plasmalogens, which are third-party verified, and there's a whole history. It's not like he invented plasmalogens. We've known about them for 100 years. But the popularity of plasmalogens will boom very soon, and you're going to see the quality of plasmalogens has to be maintained in order for this to work. So plasmalogens, again, have been in the biochemistry literature for over 100 years, but their clinical value has been relatively low because we couldn't synthesize them until 2006. And that's why most medical professionals have never heard of them, because it was an interesting thing in biochemistry, but clinically irrelevant because we couldn't do anything about it. But in 2006, the first viable precursors were created and patented via prodrome, and the results have been nothing but, you know. So this is This is an amazing video. I love this video. This comes from one of the practitioners. And the first scene, you'll see the teen, how she was acting, and that's how she would act for 12, 14 hours a day prior to starting prodrome glia. So let's start this video.
Everybody's used to making no progress in these cases.
You nailed it. That's
I,
one of the things I've seen too. Progress is like so unheard of that they are just really skeptical.
Hugely, and I understand it because I had tried as much or more than anybody,
Yeah.
I think, and literally nothing was working at all. There's doctors that have known Brooke since she was young that saw this change, and they're very interested. They want to be part of this because it's kind of unheard of.
Brookie, tell me the joke again about the kitty cats.
What kind of dog has the cleanest hair? What kind of dog has the cleanest hair? A Sampoodle! A Sampoodle? That's silly!
Prodrome glia, the omega-9
Correct.
plasmalogen precursor, is the product that seems to be anti-inflammatory
Yep.
and helps with this membrane support.
So that she's no longer a girl, but that with autism, again, she would scream for 14 hours a day, and now she's able to talk and tell jokes. I mean, that's a huge improvement, right? I mean, it's a dramatic improvement. And the improvements happen pretty quickly, like within a week, within a couple of weeks, you'll see some improvements, like Sean, 1 week on glia. But the father's a nurse, so the father was the one walking down the path, and he's tried absolutely everything. They didn't find plasmalogens until Brooke was in her teens. And now she can go out for hikes, she tells jokes, but she can also tell you what it's like to be trapped inside her mind and in her body without the plasmalogen. So it's very interesting. She they've done lots of interviews with her, and there's a lot of stuff out there online that you can follow that story. So this case involves a young boy with autism spectrum who struggled with frequent illness, significant sensory sensitivity, and delayed communication. Before support, his immune system seemed constantly activated. Maybe some of you can relate to that. Minor illnesses would set him back for weeks, and each setback made regulation harder. Sensory input, sounds, touch, transitions often felt overwhelming, and the progress in therapy was slow, and so it feels like one step forward, two steps back. From a biological perspective, the pattern suggests a brain under a chronic load, low reserve capacity, ongoing immune signaling, and difficulty maintaining signal clarity. Rather than targeting behavior directly, the focus was on supporting foundational biology, so the membranes, the immune balance, the signal efficiency. And within days to weeks of beginning membrane and glia support, changes began to emerge, not as forced behaviors, but improved capacity. And those first shifts are subtle, but they're meaningful, right? Fewer, less intense sensory reactions, improved tolerance, more consistent engagement, increased responsiveness. And as regulation improved, development followed. So improved sleep, fewer illnesses, increased pointing, right? What stood out most was not a sudden leap, but a steady trajectory forward. And so this case highlights an important principle. When the biological load on the brain is reduced, regulation can improve and learning becomes possible again. So the changes we observed are consistent with improved signal clarity, reduced immune-driven interference, and better membrane stability. So when it comes to biochemical engineering, it's about precision. It's unreasonable to assume that a balanced diet can supply everything that you need on a daily basis. For example, B vitamins, before the 90s, only 20% of people over 65 took these. And then after the 90s, after a big marketing campaign, everybody's taking B vitamins now. And the actual percentage of Alzheimer's is one of the few diseases that has stayed steady, where everything else is going up. Alzheimer's is not going up. So, and just, 30% of the population is B2 deficient as well. So this is another story. This one's, this is an example of, subtle changes. But if you have a child with autism, you will appreciate this story.
Trey, 26.
I'm Alicia. I'm 24. I'm his younger sister. I'm Kim. I'm their mom. We have a lot of laughs. Yeah.
Yeah.
But I mean, that wasn't always the case either. We've had quite a few challenges growing up. Trey was diagnosed with autism at 3 years, 10 months, and Alethea was diagnosed around that time too, around 3. Trey had a lot of sensory issues, and Alethea had more behavioral issues. So she was typically a runner. She would scream and drop to the floor, and then it would set off his sensory issues because he's got auditory sensory issues. Language was delayed for both of them. Toilet training was a challenge. Laundry was everyday, constant. Napkins were not a concept that either one of them really grasped. A lot of sleep issues. So I felt like for 9 years, I never slept because it took him hours to get to sleep. Everything had to be broken down into little step-by-step pieces. So a lot of the times I felt like a therapist and not a mom sometimes. And then about a year ago, we heard about Dr. Goodnow and the center here in Moose Jaw. And I'm like, wow, this is really incredible to have this kind of facility right in Moose Jaw. And so as a family, we came and we learned more about it, and was told that we would get supplements for free. And so that's what we've committed to, trying these supplements, the plasmalogens, for a year. Pretty much within the first week, we started seeing some subtle changes. He would stay by himself and he could cook supper.
Yeah, I cook supper.
Yeah, you're a good cook.
I cook stir fry and sometimes cook spaghetti and sometimes macaroni.
When I would go to work, I'd say, okay, Trey, I'm going to work now.
Yeah.
And often before he would say, okay, or, nothing much else. Then he started saying, okay, take care. There was one time when he called his grandma, and Trey doesn't make a lot of phone calls. Well, ever, really. I can't ever think of a time, unless he was prompted to call someone.
I called out for you when you were golfing. I'd say, Mom, where's the Thai sauce? And then I decided to call Grandma to look for the Thai sauce.
And you said, can you come and get me?
Yeah, that's what I said.
And so she took him to the store to get some Thai sauce.
That's right.
So he does problem solving now. More recently, I asked him to get me something from upstairs. So I said, Trey, can you bring my jacket? He realized, I don't have to go all the way down if I just throw it. I caught it because he threw it right at me, and he goes, oh, good catch. So these are small things, but it's unusual. And so that's what we're noticing, like a series of small things. You noticed something about Trey early on.
Yeah, I
in the
did.
first couple of weeks.
He was reading a text from someone he hadn't really talked to in forever. Typically, when he reads anything, it's out loud. Like, he can't really think about it in his head. But then I noticed he wasn't saying anything. So crazy. I've never seen him do that before in my life.
My main measure of success, wanting to be more involved with family and community and friends.
I also actually do some art, to draw and to paint.
I just would like to encourage families to try the glia, not to be scared. It's not a drug, it's a natural, it's a plasmalogen, and just to do a little bit of research and not to be fearful. I can't speak to other people's experiences, but I just know that I think there's been enough benefits that it's encouraging us to continue on. So I'm excited for the future.
So yeah, that's the prodrome glia. Now, she had mentioned it being for free. So Dr. Goodnow is from Moose Jaw, which is in Saskatchewan, Canada, and he gives all of his products out for free in that community. So if you move to Moose Jaw, you can get the plasmalogens for free just by being there. But that might be a lot to ask most people to move to Saskatchewan. But other than that, that was the program that she was talking about. But anyway, the other question that you might have is, well, can you measure levels of plasmalogens? And you certainly can. There's only one test, which is Dr. Goodnell's test, and it's called the prodrome scan. And it measures biological stress, not just deficiency. So it'll look at membrane integrity, lipid balance, oxidative stress, mitochondrial demand. I use this test a lot on all of my patients. Anybody that I can get to purchase this test, it's very, very informative. You don't need to do it. We know what's going on in autism. So you don't have to subject your child to a blood test. It's not it's not necessary. They can just start the glia. But if you want to have a biochemical baseline and track it, then this would be something that you could certainly do. Okay, and this is...
My name is Bobby. I am mother of Colt. Colt is 2 years old. He's about to be 3 in September. We have 3 other kids, so we are a busy family. I'm a real estate agent and my husband works in the oil field. He is diagnosed with level 3 autism. We didn't see any signs at all, really, of Colt having autism until September of this last year, maybe even October. It was kind of a slow progression. All of a sudden in December, we noticed he wasn't social anymore. He kind of just hung out by himself in like a corner. He lost all of his speech. He would have had about 35 words when we last went to his last well baby appointment. And then by December, he had lost them all. He had also stopped babbling, and then a lot of other common autism symptoms like eloping, a lot of head shaking, banging his head on the walls, walking on his tippy toes, general frustration. He was very aggressive. We had problems with him attacking me and other kids, lots of hitting. For the other kids, like if we go camping, somebody's always on cold duty. So I feel like for them, it definitely cuts into their childhood a little bit. We first heard about Dr. Goodnow from our functional medicine practitioner, Dr. Shelley Wilson. I just started going on like a mad hunt trying to find like who can help me get my kid's life back.
So how long has it been that he's been on plasmalogens for?
Since February.
What was his starting dose?
So a 0.5 ml was his first dose, yeah.
And what is he up to now?
He is on 2 mils before bed, and then he's doing the omega-3s in the morning. The first 2 weeks since he was on plasmalogens, we noticed subtle changes. We noticed he all of a sudden was able to have like a little bit more fine motor skills. We sent him to daycare 2 weeks after we started, and they called me and they're like, oh my gosh, like we've started trying to teach him how to build a tower, and he literally will take the blocks and stack them. And they were so surprised, like they couldn't believe it. And slowly they've been able to like build on those behaviors. He could have never done that before. He has 20 words again. And I think he might even have more. Like he tries. Yesterday, I had told you when I first got here, we were dancing in the living room, and he walked up to me and put his hands up and he said, dance. And like, I don't know, that's crazy for us because he couldn't even speak like 6 months ago. He's picked up new words and then regained most of the words that he had.
What was it like adding neuro into the protocol for him?
It got worse first and then it got better. And I remember reading that listening to that in the lectures. He had said sometimes it'll get worse before it gets better. And so when we added it, all of a sudden he was like temper tantrums. One day I was like, I've had it. Like I can't handle it. And I turned around and he was like stomping in the living room, and he picked up a dinosaur and he looked at me and he said, roar. And like he had never done any sort of pretend play at all before. And like from there, it was just uphill all the way. It actually adding that in was like, I would say a game changer. So I think starting him early was a huge benefit to him, because now all of a sudden he's behind, but I feel like he can get back on track. So my biggest, I would urge people, if you see the symptoms, to get him diagnosed early, or to get them diagnosed early. And I'm your biggest cheerleader. I think that anybody who has any sort of autism should definitely start this because it has completely changed his trajectory for the better.
So plasmalogen replacement therapy, it is research driven. It's been used in rare diseases. That's where they first used it, was RCDP. And those children, they have an inability to make plasmalogen. So they would die within the first couple of years of their life. When the plasmalogens were invented in 2006, Dr. Goodnow, those children now survive. And they are in a charity. There's like 100 of them in the world. There's not that many of them, but they're all supported through the foundation for Prodrome. But it can work in Alzheimer's and multiple sclerosis and concussion and ADHD and stroke because you have myelin deficiencies. But it is absolutely amazing in autism. So it's something to add to the work that you're doing because of all the things that we've talked about, the neuroinflammation, the glial activation, the immune system. It works on the gut, helping that the gut issues is going to be a huge benefit as well. So that's Prodrome Glia. This, you don't need a script to buy this. You can go to prodrome.com, use the coupon code PWC. And if you are interested, I work with clients around the country. I can work with your pediatrician. I can work with whoever you're working with, because plasmalogens are not, they're not well known. They're not commonly known. Most medical professionals have never heard of them, despite the fact, like I said, they have been around, or they've always been around, but that we discovered them about 100 years ago. There's tens of thousands of research studies out there on plasmalogens, their role in not just autism, but many disease states. You will see more and more of this because they're so effective, because they change lives, because it makes a huge difference. So hopefully this is helpful. This will live on YouTube, so you can go back and watch and think about it. But that biology driving behavior is a very powerful message that is important to understand. That it's not a willful thing. It is the brain coping. It is the brain doing its best in the situation that it's been given. Right. That's how it's surviving. It's doing the best that it can. So when we support the brain and we change the terrain, we can make a difference in our patients and our families and our loved ones' lives. So thank you all for listening, and I will see you in another webinar. Thanks so much.